Hey, good morning. We'll make a start. We got only 1 hour for this lecture room. Well, you all know me. I'm the director of the UWA Institute of Agriculture, and I would like to acknowledge that the University of Western Australia is situated on Noongar land, and the Noongar people remain the spiritual and cultural custodians of their land and continue to practice their values, languages, and belief and knowledge. So those visitors, this is a traditional way of acknowledging this land which still belongs to those indigenous people. On behalf of the University of Western Australia and Institute of Agriculture, I warmly welcome you to the special lecture delivered by Dr. Iqbal Khan. I got a visitor from Bangladesh. Can you just let us know your name, please? Jahiruddin. Professor Jahiruddin was the master's supervisor of Zakaria Sulaiman. So he's at Murdoch University on a visit with Richard Bell on an ACR-funded project. So welcome, special welcome to you. Now, in addition to being an adjunct associate professor, he became an adjunct associate professor at the Institute of Agriculture last year. Dr. Khan is an assistant professor in botany department or plant science department and the deputy director of the Center for Environmental and Sustainable Development at Jamia, means University Hamdard in India. It's a very prestigious university. I don't have the notes here, but it was established by one eminent person. He is the specialist in Unani medicine. You can go and look at what is Unani medicine. It's an alternative medicine. And he put a lot of money as an endowment, and that university was established. And of course, now the federal government is also funding. So it's a central university ranked very high in India, particularly in medicine dentistry. So the plant science is a small department, but it has got quite an active group there. So I met Iqbal, we'll call him, he's got a long name like me, but Iqbal in 2015 when I was the International Plant Physiology Congress in JNU, Jawaharlal Nehru University in Delhi. So sometimes you go for that. He was much young, and also I was much younger at that time. And then we talked about my talk and so on. Since then, our friendship continued. So he says I'm his mentor, but I also learned a lot from him. His research interest is on reducing stress impact through physiological markers. He has essentially targeted the staple crops, including wheat and rice, that's where a lot of the funding comes in India, to reduce their environmental stress susceptibility under the expected or anticipated climatic pressures in the future, and focused on meeting SDG goals under such scenarios. Understanding on GABA, which he will explain, mediated sourcing metabolism, defense networks, and growth and agronomic outputs under salt stress in wheat plants. GABA is largely known in the medical field, but now he's going to convince that it has got a role in plants. Dr. Khan has been honored with a Young Scientist Platinum Jubilee Award by the Academy, Indian National Academy of Science, Young Scientist Award in Indian Society of Plant Physiology, RH Dastoor Gold Medal Award by the Indian Society of Plant Physiology, and more importantly, he was listed as the highly cited research in plant animal science in 2022 and 2023, the last 2 years. So he did his PhD from the famous Aligarh Muslim University, AMU, and that's where Rajiv Washni and many others have done their PhD, very strong group there. And postdoctoral fellowship from International Rice Research Institute in Philippines. And then he worked in a few places and then finally joined this university. And he's also done some work with Yamaguchi University Faculty of Agriculture, where we have collaboration with the Yamaguchi. Today he will deliver a lecture on neurotransmitter to plant signaling molecule GABA, advancing the salt stress in wheat. Please welcome Dr. Khan. Thank you.
Excellent. Very good morning to all of you. Many thanks, Professor Siddiq, for introducing me nicely. And first of all, I would like to thank UWA in overall to invite me here and giving a chance to interact a diverse group of the plant scientists and the students. Many thanks, sir. So, as the topic of today is the neurotransmitter to plant signaling molecule. So, we are knowing that the GABA is a very significant molecule in our mammalian system and human brain system. But in my lab, we are doing mainly three things which I am going to introduce. First is that we are going to explore what the role of the plant growth regulators, especially the stress hormones in the plants, what type of the stress hormone signaling and biosynthesis regulation is going on under optimal and stressful conditions, especially in the salt stress. The second point we are looking at the lab is how the plant system is going to be taken off the defense system during the changes of the environmental stress, and how these defense systems are going to convince that you are going to mitigate, right? And third is that we are going for the more physiological and agronomical traits, and how these traits are contributing towards the tolerance mechanism in the plants, especially rice in wheat. So we are going to talk about the wheat only here. The outline I have divided into three. It is a very big project, and I am going to summarize in few slides here. So first we screen GABA for more prominent responses in the wheat in the optimal conditions. Then our goal was that how the potassium retention is going on in the wheat under salt stress with GABA. So second is GABA plus K, that is potassium plus salt. And third is that how the stress hormones are going to influence the GABA response under the salt stress tolerance in wheat. So these are the three things I am going to talk. So as we know that wheat is very prominent and very desirable crop in throughout the world, and it is the largest consumed grain crop throughout the world. And this is the 2020 data from the FAO that wheat is going to be increased, right? It's increasing, the production is increasing, although if you see the trends for the last 10 years, little bit it's increasing. But the variation of the variability rate of change of wheat crop is going very, very fast due to the climate change. And this is the alarming situation that if one day we are not going to stop that variability for the wheat production, we can face huge famine. And this is the very alarming situation, which is started from the yellow to pale yellow and so on. So this is the alarming situation where we should think about the, and you can see the major wheat growing countries are facing the alarming situation, right? So here we come to the climate change. There are so many things, lot of things are in the climate change. High temperature is going on, CO2 concentration is going on high, but the salinity is the major problem in the agriculture soil due to the various factors which we know very well. The population coverage of the fortified salt with the mandatory fortification is like that. So this is again the alarming situation. The major wheat growing countries are facing the salinity problem. So as a consequence, we have worth $27 billion US annually have reported in the salt affected regions. This is the loss, and this loss is directly from the farmers. The farmers are going through the very hard time to do the economic loss, and this need the sustainable approach which can reduce the impact of the salinity stress. So if you are going to see a holistic approach for the sustainability wheat production, so we can found that we have phytohormones and mineral nutrients, right? So these are the two major factors which are going to which are going to be part in the salinity tolerance, and this is our focus. So now GABA. What is GABA actually? It's a non-proteinogenic amino acid, and it is very prominent in inhibitory neurotransmitter stress reducing, sleep enhancing effects, and it helps in the reducing the anxiety, stress regulation like that, and blood pressure. It maintains the blood pressure. These are the story from the mammalian cells, the human body, right? But it was around 1949, there was one paper in Science, and they reported the GABA for the first time in the plants, in the potato, right? And after that, so many literature we are going to see that the GABA is accumulating within the plants, but what role is going to be play during the stress responsive plant. So plants also communicate using their own kind of nervous system. So is GABA is also playing a nervous system role? We need to talk on that. We need to look into that. So this is a very brief journey of GABA as a plant signaling molecule, which is starting from 49, and the major outbreak comes that GABA accumulation was affected by the stressful environment in 1995. Then 1996 we discovered about the biosynthesis. More biosynthesis accumulation is going on through the GAD. In 1999, the herbaceous thalina grows efficiently with the sole nitrogen resources. So this is the first evidence that GABA is underplay with the nutrients. Sorry. Then 2020, there is a lot of gap. We observe with the GABA POP1, 2 and 2, 3 mutants. I will show. And after that, we started for the environmental assess impact and GABA inhibitory effects, right? So screening of wheat genotypes to categorize salt tolerant and salt non-tolerant genotypes we have done. Studying what dose is going to be play an important role for inducing the defense system and stress hormones and all the things, and what type of the interaction the GABA potassium has to reduce the salt impact, and how the GABA is going to interact with the stress hormone, that is ethylene. So same thing. So we screened around 15 wheat genotypes. All are high yielding in nature, more than 4 ton per hectare from the Indian lines, which was created or manufactured by the PAU, Punjab Agriculture University, or IRI, and we screened under the different regimes of the salinity, started from 0 to 200. 200 is too much for the wheat, but we were interested in how the very high salt tolerance is going to affect that hybrid, high yielding wheat varieties. So we found that the GABA accumulation is very prominent and very variability, and we found that the line number 11 have showing the very much GABA accumulation at the all concentration of the salt treatment. On the basis of that, we categorize into 2 tolerant and non-tolerant lines, and we also observed that the all localization of oxidative stress markers, and you can see that there is a very prominent distinct between the oxidative stress markers in the both genotypes. Then we went for the differential concentration of GABA test in the wheat, the same line, the tolerant line, and we started from 0 to 3 millimolar dose, and we found that 2 is inducing very good height and plant dry accumulation. The chlorophyll content was very good and the Robisco activity was very good. There was so many data, so I am just showing a very brief, the very key data. Then we went for the biosynthesis, GABA biosynthesis inhibition through AOO. So we started from 0 to 10 millimolar, and these doses are picked up from our previous data. So all concentration inducing significantly the GABA accumulation and its enzyme, key enzyme. Stomata was the key because we are also interested in how the GABA is going to influence the stomatal response and how a stomatal response is going to be associated with the potassium, right? the plant dry mass was reduced significantly, suggesting GABA is playing a very good role. So we went further with the mutants of the GABA biosynthesis, and these mutants are the GABA T, that is transaminase is a key enzyme which are going to play a very important role in GABA synthesis, and these mutants show very significantly the phenotypic. You can see the POP21 mutant that is knockout, and POP2 knockout gene is very promising, and size of silicon is reducing from the wild type, leaf size is very less, flowering time decreased. So all are suggesting that GABA is playing very important role in growth and development of the plants. So again, the mutants show very less activity of the GABA T, GAD activity, number of silica reduced, number of seeds per silica reduced, GABA content is showing very variability and with reduction in the synthesis, right? So these are the GAD mutants which showing the stress markers under the salt stress. Root cell viability is showing that if you are going to reduce the GABA biosynthesis, you will get more accumulation of ROS, less phenotypic representation, right? So less phenotypic growth is going on without the GABA biosynthesis. So again, we tested the alone the GABA plus salt stress, and you can see that nut photosynthesis is going to be affected under the salt stress. If you compare this data, you can see that the salt under the salt stress, GABA is protecting photosynthesis, maximum PS2 efficiency, stomatal conductance maintained, internal CO2 concentration maintained. The most important, we are retaining the crucial nitrogen content with the help of the GABA. Sulfur is also maintained, and as a result you can see that our plant height is protected under the salt contaminated soil, and the plant dry mass is going to be very good in accumulation as compared to the salt affected wheat. These are just a phenotypic representation. So another story is that how GABA is going to associate with the any nutrient such as potassium. Potassium is very important in the salt stress. If you can retain the potassium during the salt stress, you can achieve your goal, right? The idea was that if you are going to protect the potassium in influx, then you can make a salt tolerance plant. So we tested with the few treatments, GABA, potassium, and the combination of both under the salt stress and alone. And you can see from the RS accumulation, the GABA plus NaCl less, K plus NaCl less, and if you are going to use these both traits, very less, right? So this is the hydrogen peroxide and localization of these molecules. Same conditions with the superoxide radicals, same with the other oxidative stress markers such as methyl glycol content and lipoxygenase activity, right? Same with the TVARS content and cell death visibility, you can see the very prominent in the treatment, right? And your growth protected with that treatments, and the root shoot toxicity tolerance index improved, right? The salt tolerance index improved with the help of potassium and GABA under the stress condition. Stomatal frequency is also going to be very protected during the salt stress condition with the potassium and GABA, and the more protection was given by both together. The structure, you can see very prominent and very influencing with the stomatal aperture. The yield was protected completely, and this is the most desirable thing if you are going to protect the yield. under the such conditions with the help of any molecule and any nutrient, especially with the GABA and potassium. So you can see the yield was very much protected, the spike length per plant was protected as compared to the salt induced, right. So these are the representation of the stage 45 days after sowing, 75, 150, and the increase or decrease is going on with the help of the GABA and potassium. So in conclusion, you can conclude that our GABA and potassium is cumulatively working for eradicating the salt stress and reducing the oxidative stress, increasing the defense system, osmolalite production, although I did not mention here with the data, nitrogen assimilation, carbohydrate photosynthesis, all the traits were protected very well during the salt stress. So another story is from my lab is how GABA is going to be correlated with the stress hormone. I am going to just tell about the ethylene, although we are working on ABA also. So few data is not here, convincing data is not here, so I am not going to tell. So GABA and ethylene has few reports earlier, and they were saying that yes, they can influence each other to protect the stress in plants. And to test that, we use lot of combinations such as GABA alone, ethylene alone, salt stress alone. All the three factors were alone. The combination of GABA with salt, ethylene with salt, GABA with salt, and ethylene salt. And we used the AVG, the biosynthetic inhibitor of ethylene, and the biosynthetic inhibitor of GABA is AOA, which we showed earlier, right. So you can see that there is so much influence of GABA and ethylene together. You can see if GABA ethylene salt, see the difference between the oxidative generation is very prominent and very well. Same in the case of other oxidative stress markers, very well protected with the help of GABA and ethylene under the salt stress. Cell death is very low now with the help of GABA and ethylene. You can see ethylene, GABA is very less. See the difference. It's a very promising data, right. So we did for metabolic profiling how the defense system is going to be regulated with GABA and ethylene, and we found that under the control condition, as normal, the defense system is not well regulated, but with the salt stress, with the ethylene and GABA, the antioxidant system and few nitrogen compound which actively which expressed very highly, such as that 1,3-chlorophene 5-phenoltriazonamine. This is the most abundant molecule with the help of GABA application. So in the presence of the biosynthesis, we found that those antioxidant system was suppressed. It means that GABA is not alone playing a role. It means that ethylene is promising, inducing, and helping the GABA into induce those type of the compounds. So again, we were so much interested in looking for the stomatal structure and the stomatal data. So you can see that these are all the same images, and the most prominent is in the ethylene plus GABA. The sulfur content was very much influenced, and you can see that GABA plus ethylene is going to be protect more, alone less. The serine, the all sulfur assimilation pathway is going to be protect, well regulated. These are the few representation. You can see what type of the growth done by the GABA plus ethylene, right. Yes, in conclusion, we can conclude that the GABA is very prominent in protecting the salt stress in the wheat, especially through multi-channel. Oxidative osmotic stress, oxidative stress going to be lowered, uptake of ions that especially the potassium ions is going to be optimized, and morphological alternation is very well prominent. These all factors are going to contribute towards our yield stability. Yield stability should be our aim, so we went for that yield stability also. So these are my group, and I would like to thanks my all funding agency, that is UGC, DST and DBT. I would like to thanks the university, the Jamia Hamdard, also the UWA. I'm open to all. Thanks.
Thank you, Iqbal. He went very fast, and so we have got a lot of time, but I'm sure that people will have some questions to ask to Iqbal. Who want to ask first? Okay, Zakaria.
It's a very nice talk.
Thanks.
My question is related to potassium.
Mhm.
It shows that potassium and GABA contributed almost similar, but when you combine,
Mhm.
it become less.
Which one?
It become less the growth. Is it true?
No, it's not
Can you
like
just
that.
show quickly?
Yes.
Potassium and GABA. So, because you went very fast, some of those slides.
Oh.
Maybe a reduction in certain stress hormones. That's what
Stress.
it's
Actually,
already-
you saw in the salt stress, I think.
Yeah. So it would have reduced the stress hormone. So that's what it was
Mm.
very fast, but
Oh.
Yeah.
Well, I didn't see the time actually. This one?
Yes, it's the pot experiment.
yeah.
The potassium alone.
This one?
Yeah.
But you see the baldness of the leaf.
Yeah, but look at the data and show that. Yeah. So
Yeah, but
that
data is okay. The root length is good because we have the salinity stress also, right? So I'm claiming that this is not going to elevate completely. It's going to protect, right?
Yeah, because,
So it's
here it is
protecting.
potassium alone.
Yes.
But then you need to look at the sodium chloride.
Yeah, we have to compare with the stress treated, right? NaCl, because you are not going to like that. Okay, this is the miracle, and this is the see the prominency. Very less in number of the leaves, and the leaf number is going to be increased, and the chlorophyll content like that all are going to be increased as compared to the salt stress, right? Yes, we are comparing to the salt stress, right? Yep.
Any
Thank
other
you.
questions? Yeah.
Yes, please.
Shen.
Further to this slide, GABA and potassium, it seems GABA and potassium show similar level of function in response to salt stress.
Mm-hmm.
And combined treatment with GABA and
Potassium.
almost double effect
Mm-hmm.
in some
See.
cases.
Yes, because you know potassium is regulating their own metabolic activities, and GABA is completely their own. If you are going to merge both heroes, so it will be like, okay, this is the benefit for the plant under the same condition, right? So condition is same. We are testing GABA alone, we are testing potassium alone, and they are doing very good role alone. But you are going to combine both, so obviously they will be inducing their own metabolic activities, and the response will be like very good.
Yeah.
So this
So
is the
this is very interesting result.
yes, really,
I have a silly question.
yeah.
Because GABA and potassium has similar direction
Mhm.
for the in response to salt
Salt,
stress.
yes.
My silly question is,
Mm-hmm.
how about if we double the concentration of GABA itself or potassium itself? What's the effect?
What's the effect? Okay, you can see the GABA and the potassium. The shoot plant height is more in potassium, little bit. You can see, and GABA have little bit less. But GABA plus potassium plus NaCl. Remember that we have the stress condition now, right? The both is going to be like that, and see the response is very less, but more than the NaCl alone. So this is the aim, this was the aim to achieve how these are going to protect under salt stress condition. The alone condition like a GABA and potassium in optimal condition is not a concern, because we know that if in optimal condition you are going to supplement any fertilizer, any molecule, anything, that will be enhanced and that the growth will be enhanced. It's obvious that, right? But remember that during the stress condition, what type of response we are getting, and we are getting as compared to the salt stress with both. Yes.
So in this case, you use a certain concentration of GABA.
Yes, we screened, nah?
Yeah.
We screened
But
earlier.
if we double the concentration.
Yes, yeah.
So what he's asking is, one, you screened GABA and found that 2, if I
2
remember
million,
correctly,
yeah.
and you screened potassium, probably, we know the rate, and then what he's saying is that when you combine, you haven't looked at a rate response, maybe worth looking at.
Okay,
That's what
that's
he's
great.
saying. That's what
great.
Prashant
Yes,
is
great.
saying.
Oh, thanks.
Now, I got a question that, so let's say all looking good, how do you apply? Is it a foliar spray? At what stage? And how potentially we can take into a farmer's field? The farmer
Yes.
got so what is your strategy there?
The idea of applying the GABA and potassium is a fertilizer, right? So we are mixing in the soil,
So,
right? The GABA is a new molecule and we want to be a very commercial level. So whatever the farmers are going to be foliar apply in the field for the growth and conditions, we applied for the foliar application of the GABA. So keeping in the mind that, so in the future we can use that GABA concentration to the whole field, right?
And what will
Yeah.
be the cost approximately? I mean, it's
Yeah,
not
cost
yet out
cost
there.
is very less.
Mhm.
Cost is very less. We purchased in Sigma that GABA molecule for 10 gram. It was like it was in Indian rupees around 3,000 rupees.
Cheap.
You can convert into the like $60,
Mhm.
60 Australian dollar
Mhm.
for 10 gram.
10
So very very cost
And
very
10
cost
gram will
effective.
take up how many hectares?
Yeah,
Approximately?
it can. If you are going to, we are still using for 2 years that 10 gram, right? So we can calculate further so on. So it will be like
But you haven't tested this in the field yet.
not yet,
No.
but this year we are planning to collaborate with the IRI
Mhm.
for some field experiment, and we will be testing in the field also. So this.
Is there any available data that regarding GABA effect on another crops like rice or?
Yes, in rice there are so many data you can find, but more than wheat and rice I never seen. Arabidopsis is there obviously, but rice is getting much attention now. Yeah.
Zachary again?
Do you have any research, I don't know, on zinc?
Zinc? No, I did not test any.
May play a role.
Probably.
Yeah, maybe, because it is all about the essential nutrients, so we can try it.
But under salt stress, I think
K is, yeah.
very, very important in terms of cations. Yeah. Eric?
Why do you think that a molecule like this hasn't been evolved in plants?
Molecular?
A molecule like GABA
Mhm.
is not evolved in plants. It sounds like a miracle.
Yeah, actually yes. We are doing a lot in the plant science, but the GABA molecule was first detected in 1949, right? So after that, I don't know why people neglected it.
The question is like this. So, for example, let's just put it in this
Yes.
way. If I go and look at it all over the world, I can collect wheat germplasm from salt-affected area. And they have evolved and adapted, if you believe in Darwin, yes, they have adapted, they have a tolerance. So if I go and test a land race from a salt-affected area, which we can give you from the germplasm, and then see whether they have developed GABA, that is the question, correct? Something like that.
in wheat. but
In
also-
wheat,
also
yeah, just in
plant-
other crops, yeah, in
plant
other plants,
kingdom in
yeah.
general.
Yeah.
Okay, so fundamentally I cannot comment on that
Yeah.
because I did not went from that point. So maybe we
Maybe
can,
worth looking
maybe,
because
yes,
India has got
maybe.
huge genetic
Huge genetic
germplasm in
diversity.
their collection, and either rice, for example, some
Yep.
rice is originated in the state which I was born, tolerant to called Pokali,
Called Pokali.
tolerant to salinity.
Salinity.
And that gene was now used in IRI for two things. One is for submergence
Yes.
and so on. So maybe worth looking at it.
Yeah.
Is there any
Saltol.
naturally originated? That's the
Saltol one.
question. But these are synthesized GABA.
GABA,
you know,
yeah.
sigma
Yeah,
is-
this is a good idea, and I will keep in mind for further molecular and all
I
those things.
suppose the other side of that question is that generally when you don't see these
Mhm.
compounds naturally occurring, it's because there's some trade-off, right?
Yeah.
There's some negative aspects
Mhm.
of it.
Mhm.
So I could imagine physiologically what you're doing,
Mhm.
you're encouraging certain processes to protect the plant, which may make it more susceptible to something else, and
Mhm.
that's why it's not occurring naturally.
Mhm. Maybe.
But it looks
Maybe-
like
maybe.
it's not giving any, in this case, no negative effect at all, right?
Yep.
So if you look at just the plant height GABA alone, but that protection which you're getting is remarkable.
It's remarkable.
Yeah.
Yes.
Yes. Any other question? Yeah.
So I'm curious about how GABA gets ABA.
ABA.
So, yeah.
Actually, we are doing on that, and we have good data from metabolomics and all things. So I will be going through that data. Then I will let you know. Because ABA and ethylene both are a stress hormone, but they shows very different way. Yes, because ethylene is a gaseous hormone, and it can influence some senescence process, some photosynthetic process. But ABA is significantly influencing directly to the photosynthetic attributes, especially through the stomatal conductance and all things. So we are looking in that, and the good data is there. So more stomatal response was observed during the ABA and GABA response, instead of that nutrient like N retention, potassium retention. So we are digging on that data actually. So maybe after 1 or 2 months we will be communicating some good journals like that.
Any other questions? If not, the other one is high value crops.
Yeah.
For example, you've got quite a lot of salinity, secondary salinity with irrigation and so on.
Mhm.
So you've got things like broccoli and many other crops. So you get immediate response and
Response.
economic returns.
Yes.
Because things like rice and wheat, okay, but the problem is leafy vegetables, some of the fruit trees, they are all very susceptible.
They
So
are.
if you then in an orchard and things manage that, then you may get a different response. I'm just
Yeah.
suggesting. yeah.
In this way we can establish the GABA as a plastic marker. Yes.
Any other one? No? All right. Thank you very much. I'll just wrap up. Please join me in thanking for the wonderful talk. And if anyone wants to meet, he's still around for the next couple
Yeah.
of days. And I'm sure some of you are already meeting. Yeah. So before I bring to close, I would like to inform that Institute has a number of fantastic events on the horizon. One is the important one on can Australian agriculture achieve net zero? It's at the University Club Auditorium. Minister for Science is coming, and number of speakers. You have the QR code, and some brochures are there. So please join, particularly students. Try to encourage. This is a big topic at the moment. Can we achieve that? And there will be keynote speakers and panelists come and discuss that. Then we also have the Shenton Park Field Station Open Day. This is just about 4 or 5 kilometers from here. We have a 65-hectare property there, and there's quite a lot of research going on there. That's on the September the 13th, 2024. Again, keep an eye on that. We normally do it once in 2 years or 3 years, so there's quite a lot of excitement there as well. And then we have the postgraduate showcase, which is next week, and already it is full, 140 people, etc. But so we don't want to advertise widely, but some of the postgraduates in particular and their supervisors should come. Is that all? Yeah, thank you.
If you follow the QR code, it takes you to our Eventbrite.
Yep.
There are a couple of other lectures that
Yeah, there's another one coming on
28th of May, we're doing one with the School of Agriculture and Environment on the value of postgraduate studies, studying internationally. And
talking about
Eyes in the sky.
eyes in the sky, basically satellite technology, how that can be used in terms of understanding adaptation of the plant, drought. So he has got a lot of experience. He published a book. Some of you may have seen him at the morning teas and Adjunct with us. So with that, once again, thank Iqbal for your presence, for your presentation, and all of you to take time out and come and listen to that. Thank you.